Welcome to Strategies for Fresh Cut Mango Preservation. My name is Cesar Favero, and I am the Manager of Relations with the Industry. I'd like to introduce you to our speaker today, Dr. Francine Kukel, who is a full professor and researcher at the Federal University of Paraná, where she teaches undergraduate courses in agronomy and master's and doctoral courses in agronomy and plant production. She teaches horticulture and post-harvest. Her research focuses on post-harvest of horticultural products with a view to increase the shelf life of fruit, vegetables, mushrooms, and ornamental plants. She obtained a master's and doctorate degree at ISALC, University of São Paulo, and her doctoral research at the University of Florida in the United States, as well as a postdoctorate in post-harvest at the Instituto de Investigación in Spain, IRTA. Professor Kukel, welcome. Thank you, Cesar. Thank you to the Mango Board for this opportunity to be here today. It is a pleasure to be in touch with so many producers, the industrial sector, as well as any other participants who may be here today. I hope to fulfill your expectations and provide the information that interests you, and I will be at your disposal at the end of the presentation to answer your questions and also by email. This is the presentation I have prepared for you today. Strategies for fresh cut mango, insights for the mango industry. I would like to begin by presenting a brief agenda. First, I will cover the factors affecting fresh cut mango quality. I would also like to talk about changes that happen due to processing, enzymatic browning, microbiological aspects, and new strategies to preserve minimally processed mangoes. As I believe most of you know, the specific cultivar selection is among one of the most critical practices to have good quality minimally processed mango, as well as pre-harvest agronomic practices, ripening stage, the quality of the uncut fruit, storage conditions, processing, preparation methods, packaging, storage conditions are all very important to preserve quality. Now I will go briefly into how these conditions affect the product. First of all, it's important to mention the key varieties or cultivars that are used for minimally processed mangoes. Kent, Keat, and Tommy Atkins are the key varieties. Kent is preferred due to its lowest fiber content. Consumers do not want to consume very fibrous mangoes. And in addition, any mango with a higher yield of fruit tissue will be preferred by the market. Larger size fruit are usually preferred as well for processing, but I also must highlight that these cultivars are the most well-known in our region. However, there have been research done in other countries with other cultivars, which I very well may refer to during my presentation. As to agronomic practices, it is known that post-harvest processes are unable to improve the quality of a product once it has been harvested. And this has to be made very clear to the industrial sector. It is important to produce a quality fruit. It is very difficult to preserve its condition or lose quality. So the key focus should be at the orchard. And this is the main point of my presentation. There are multiple research projects on pre-harvest of minimally processed mangoes, but we find very little data about what conditions should be provided so that my minimally processed mango does satisfy consumers' needs. And that is the question that I would like us to save for a later discussion. It is very important to focus on how to manage production itself at the orchard. And one of the important elements of that management has to do with calcium content. Now, going into ripening stages, that is also very relevant for post harvest quality. If a mango is harvested too early, what we will find is minimally processed mango that offers low quality. It's impossible to harvest a mango when sugar was low, acidity was high, management was inadequate. I will not be able to correct those factors post harvest. So, that is not a stage that we recommend for harvesting because the mango had not reached the ideal ripening stage. It is ideal when I have the ideal concentration of sweetness and acidity so that I can satisfy my consumer sensory demands so that we are fully committed to the quality of product being delivered. When we go into stage 5, we see it is not a recommended stage for harvesting. Since the mango, the fruit, is more ripe than it should be, it will be very difficult to achieve a minimum 10-day shelf life as typically required. So the ideal stage for harvesting is stage 4. But I should point out that among different cultivars, we will see external differences in terms of color, size of the fruit, tissue, texture in stage 4. So it's important for producers to know exactly what is the appropriate stage for harvesting so they can send their product to the industry according to each cultivar. And the industry should know as well what product they need so they can educate producers. So this dialogue between producers and the industry is key for a quality post-harvest product. As to the quality of intact fruit, uncut fruit, in order to maintain quality post harvest, producers need to have good practices so that the product is safe for the consumer. Likewise, traceability is important because it will allow the industry, if there are any issues, they can quickly reach the right producer and find out what happened out on the field so they can pinpoint the origins of the problem so they may not happen again. And psychochemical features are also very relevant so we can satisfy consumer demand and preferences. I should also point out that since mangoes are produced worldwide, there might be different preferences according to different consumer choices. Each location will have specific requirements. And if the industry is not knowledgeable of what their local consumers prefer, we recommend that research be done so that they can find out exactly what consumers are looking for. Now, going into processing and preparation methods. All conditions that will foster quality in terms of food safety practices are essential, both in regard to storage, processing, and preparation. There is extensive literature on these topics focusing on a specific care on sanitation and safety practices, as well as labor training, conditions within packing houses, compliance, inspections, which should be taken seriously by producers and the industrial sector, as well as storage conditions are so relevant. If mangoes are harvested slightly prior to ideal ripeness, it should be stored at between 20 and 22 degrees Celsius. Once it achieves ideal ripening, goal temperature would be 40 to 45-degree Fahrenheit because cold temperatures can hurt the fruit. It would be injured by chilling. So please remember these temperatures. -4 to 7 Celsius, that will harm the fruit and affect some of the safety conditions. And humidity should be 90 to 95%. What changes mangoes undergo during processing? There will be biochemical changes because as the product is prepared, it will be peeled, which will disrupt the fruit's integrity. And what do I mean by that? It means it will physically disrupt cells, which will cause a series of biochemical changes, some desirable, others not so. For example, when I disrupt cell integrity, I lose integrity, which means that the mango tissue will soften. If that happens to a high degree, shelf life will decrease. Another change is that once I disrupt cell integrity, I lower respiration rate, which is inevitable. And or rather, I increase respiration rate, which means I increase the intensity of every other biochemical senescence process, which leads to lower quality. So I need to minimize those changes so that my product can at least reach 10 days minimum shelf life. One of the changes also caused by the increase of respiration rate is the increase of ethylene production. And ethylene induces senescence, which means the higher amount of ethylene is released, the lower the overall shelf life. So as senescence advances, I lose water and I may not have a visible problem in minimally processed mangoes, which is enzymatic browning. So, what do I need to do to minimize these processes? It is inevitable for a mango that has been processed that eventually will have to be peeled, which will advance the process in terms of biochemical changes.
And one of the main biochemical changes is the increase of the production of enzyme, which is called polyphenol oxidase. The more active this is, it will actually create a product called quinone, which is a product that is developed by the tissue itself that, as you see on the photo, uh, makes the tissue darkened. So it damages and gives the appearance that this is an old product with no quality. So I should reduce the production of polyphenol oxidase to avoid this enzymatic darkening so that the mango does not look like it's of bad quality. What other causes do I have of quality loss and this enzymatic darkening? Some cultivars are not appropriate to make minimally processed mango, basically because this polyphenol oxidase is very intense, and I touch the mango and it gets darkened. So the conditions of warehousing, the suboptimal conditions of warehousing, the orchard, etc. All of this is important. So what I would like you to understand from now on is that as much as I have a very well-regulated process from the peeling of the mango, I increase the shelf life of the product. Any failure in this process of a suboptimal process will decrease the shelf life and durability less than 10 days of this product. So when I mentioned a while back about the fertilizers of the orchard, and I mentioned that the main studied factor until today, or in other words, the only one studied until today, is calcium. And why calcium? Because this is the element responsible for the cell firmness and actually the cell integrity as well. So you can understand better. This is a cell wall, and it's in this cell wall we can see microfibers with cellulose, middle lamella, and other components in the cell wall that are interconnected by what? By fibers, pectin fibers. And these fibers work as if they were a string that ties together all these fibers that actually create the wall. If I have little calcium, I will not have enough pectin to tie together these fibers. So when we harvest the mango, the skin and the cell wall is less strong, which causes that when it's processed, the injury by the knife, the processor, the dryer will cause in my fruit will be much higher. So it is essential that in the field, I introduce calcium to produce pectin. And when producing the pectin in this way, I can interconnect the microcellulose fibers. And this information is important to mention, that it's not just to know that I need calcium, but I need to know how much calcium to apply, when to apply it, how to apply it, because calcium is a chemical element that is not very mobile. So it has to be moved to the fruit. And if it is not transferred to the fruit, I will not have the cell wall to deal with the handling. When can we do this application? It can be done in the soil. It can be foliar application or post-harvest application. We will see this with more details next. In this slide, we see the enzymatic control of the browning in fresh cut mangoes with the application of its effect of this polyoxidase enzyme in the treatment water of the fruit, in this case of calcium. What happened when I applied calcium in the water with which I treat the fruit? This is the activity of the polyphenol oxidase that was much lower than when there was no calcium in the water. Therefore, it is important to add to the treatment water calcium to increase its durability. And here we'll see the effect of the calcium in the browning of the fruit and the effect of the calcium in the classification or the score given to our product. So this is with only water. The darkening, as you can see, throughout the warehousing days, there was an enormous darkening, more than the double than when we actually applied calcium in the treatment water. And when we see the general appearance, I also see that in terms of general appearance, at the end of 6 days, I have almost 50% more score to this fruit. And when we look at the fruits here on the picture, what I see is that the sensorial analysis after the calcium application registered a much lower darkening. This is enough for you to actually evaluate the possibility to add calcium as a vehicle, or calcium that is soluble, that adjust the dosage regarding the cultivar you're working with. So everything has to be carefully handled to make all elements work. So here is the effect of the calcium lactate, and here we show other conditions, the effect of the application of calcium in keat mango. And the calcium that I see here, which is the interesting for us, 1 or 2% vis-a-vis control regarding general appearance. You can see the general appearance of the control. There is a classification 3.8, and when calcium was applied 8.33. It's obvious the difference between them. It was evaluated in 2 harvests, and in the second, we also see significant differences showing how essential it is to apply this calcium. Here, regarding or the relationship between sugar and acidity, this relationship shows the probability of this fruit to be more flavorful. When it's something that we see different letters, we see that statistically the values are different. So when we focus on 2% calcium vis-a-vis control, even though in numbers the difference don't seem very stark, statistically they had scores that were quite different from B to D. So this tells us that the products were indeed very different. In the same way, in the second harvest, proves the relevance of these. So talking about microbiological organisms. I talked about the metabolic changes, and now will speak about microbiological organisms. I cannot say which one is more important, but I can say that regarding the safety of the food, they are essential because there are many microorganisms that are in the surface of a mango when it's harvested, like Escherichia coli, Salmonella, Micrococcus, Staphylococcus, and many others, as you can see on the screen. And I would like to recall what I mentioned about the ideal temperature, not to cause any lesions to the mango, 4 to 7 Celsius. If I go below this temperature, I will have an injured mango, darkening process, and loss of quality. What is then the optimum condition to reduce the activity of these organisms in any product? It is temperatures close to zero. If I have a product contaminated with any of these MOs, and if I maintain the mango at 4 Celsius, the uh, colonization of these microorganisms is very fast because many of them are uh, detrimental to health, sometimes actually very serious bad and nefarious effects to health. So population is highly connected in regards of the importance of not consume contaminated products. So this is the question. If one microorganism goes through the improving process, how will I decide to maintain at a low rate, maintaining it at a temperature of at least 4 Celsius. This is the importance of the industry to know that they have to work with a level of contamination of any microorganism close to 0 Celsius. So in addition, make treatments so if there's any microorganism survives, it is still able to control it during processing. And that's what we're going to focus on the rest of the presentation. What are the main causes of contamination of a fruit? It is the external field contamination, the facility sanity contamination, the washing, rinsing water quality, and the pathogen internalization due to the high or tepid temperatures of washing rising water. So the temperature with which I treat the mango favors that the pathogenous element that is on the outside enters the fruit. And if it enters, the control is much more complicated than when it is only at the surface of the product. How can we normally control these microbiological organisms in the mango? The thermal technique is the most used, but what is the problem of the thermal technique? When I apply temperatures for a minimally produced product, I can cause the softening of the product, loss of flavor, loss of firmness, and all of this results in the loss of quality of the product. Reason why we need to test other methods, which were the new technologies recently developed that I will present now. Because other techniques that are already in focus that compete with this thermal technique, it's irradiation, antibiotics, and chemical control. Until recently, there were not many concerns from the population, but recently the population does not tolerate the usage of these products because of the effects on health. So the new technologies are called non-thermal, and they do not use chemical or products or antibiotics or radiation. So these techniques are considered safe. So these technologies are pulsed light, LED or light emitting diode, cold plasma, neutral electrolyzed water, and acidic electrolyzed water, and high intensity ultrasound. These were the techniques that I found within the spectrum of mango industrialization. Very few mentioned, but I found them. There are some techniques that are cited for other cultures or harvests, but never for mango. So I'd like to highlight that the need of information, scientific information, the lack of investment in research has resulted in the lack of information about these techniques used in mango. We know about the usage in other species, but very little in mango. And the little we know, I will present, but I would like to explain that all techniques were tested for one cultivar, but not for another, for one type of planting or seeding and not another. And each producer should test in its own condition, each in the industry, so that the process works within its totality. So let's see how the mango has been treated to avoid microorganisms using uh, sodium hypochlorite and calcium hypochlorite. These are applied in pH of 6, 2 to 5 minutes. This is an established industry method. However, when this hypochlorite touches the organic matter, the organic matter comes from where? The fruit has in its own surface this organic matter that it brings from the field. The problem is that there are reports that this can produce chemical compounds like chloroform, trialomethanes, teratogenic allostatic acids, and they are carcinogenic. And this shows that industry, increasingly so, has been asked to replace this process for a process that is safest for human beings.
When the same substance is applied in an alkaline pH along with nitrogenous bases, we also have the possibility of generating carcinogenic compounds. So, because of that, the FDA set a maximum residue limit of sodium hypochlorite at 0.01 milligram per kilo of product in the United States. Beyond that level, the product becomes harmful. There's a risk of harm to human health. So there has been a search for alternative methods for microorganism control. The first one I will show you that has been used after hypochlorites has been the application of ultraviolet rays on the product while it's being processed. What happens is that ultraviolet light can have a germicidal action, killing microorganisms on my product. Peak efficiency is around 254 and 262 nanometers. And what does that UV light do besides killing microorganisms? It promotes the production of ethylene, of ABA, of phytoalexins. So it also causes physiological effects on my product. Some of them are quite favorable. For instance, it delays ripening or the softening of the fruit tissue. But some other effects are terrible because it reduces the overall weight of the product. So there are positives and negatives. How can we minimize the negatives? We need to have more research to find the ideal exposure for each product. Here we have a picture showing fruit being treated. This is not an example of mangoes being treated, and we can see the ultraviolet lights. What is the main issue of UV light? The issue is mercury, and society rejects the use of mercury because it is a pollutant. So, among pros and cons, we have the positive of it being approved by food control agencies. It is a strong germicide. However, despite causing relatively small changes in physiochemical qualities, it is harmful to operators if they are not properly protected, and consumers show concern because non-ionizing radiation is produced. So we have seen two techniques so far: hypochlorite, which generates carcinogenic agents, and UV light, which can also be harmful to those who handle it. It is potentially a pollutant. So this is a difficult situation to resolve. Now I'm going to present to you an example of use of UV light, which allow to achieve a shelf life of 12 days for Tommy cultivars of mangoes, which is a wonderful result. However, the International Agency for Research on Cancer does consider it carcinogenic to humans, even though it can generate production of carotenoids, phenolics, flavonoids. That concern does not make it a viable option. Now let's look into new strategies for minimally processed mango preservation, which can be used in all of those products so that it is free of contaminants, which are not visible to the naked eye. However, it can bring multiple risks to consumers. Among these strategies, we want to have a tasty mango. We want it to protect both operators and consumers' health. We want it to be clean and safe and eco-friendly. The first method I will present is pulsed light. Xenon is a brand. It is a company that produces this kind of pulsed light. Just to give you a general overview, light is composed of multiple spectrums, the X-ray spectrum, all the way to infrared spectrums, all the way on this end to the right. Each spectrum has a specific function in terms of controlling microorganisms as well as medical functions in affecting various parasites, virus, bacteria, fungi. Pulse light is similar to UVC. However, there is no mercury involved, which it is far more eco-friendly. Traditional UVC has to be applied for minutes to hours. However, pulse light is only applied for seconds. It is a very short exposure time needed. Lamp pulses can be applied for milliseconds depending on microorganisms that we want to remove. The human eye cannot even detect the pulse light being switched on and off because exposition can last only milliseconds. However, its intensity is quite high, 200 times higher than the sun at the sea surface. One of the advantages is that it preserves nutritional quality, and it has been approved by the FDA. It was approved back in 2017. Here we see Listeria monocytogenes, and we see here the wall damage caused by pulsed light, and it is removed, and not a trace is left on the product. Here we see APL equipment and how it can be applied on apples. That's an untreated fruit going through roller conveyors. It is exposed to pulsed light. And as I said, the amount of heat is quite intense. So it requires air circulation in order to remove that heat effectively. There are lamps provided by the brand Xenon. However, there are other competitors in the market, and each wavelength will produce a different result. For a certain cultivar, perhaps Xenon would be best, and for another, perhaps Krypton would be better. And what are some of the critical factors for this process? The distance between the product and the light must be optimized, as well as the distance between the input and the output, volume, geometry, orientation, whether we want a parallel or a diagonal ray. Here we have an example developed by Charles et al. In 2013, showing that applying 4 pulses of 2 joules per squared centimeter, which is equal to 8 joules per squared centimeter per sample, allowed for storage for 7 days at 6 degrees Celsius. So what is the advantage? Since we know that a mango can last up to 10 to 12 days. Well, for that to happen, temperature would have to be lower. So perhaps I have a benefit related to the temperature for storage. Perhaps my retailer is not able to store mangoes at a lower temperature. So by allowing for storage for up to 7 days at a higher temperature, perhaps now I can sell my product to different retailers who don't have that lower temperature available for storage. Now we are going to move on to a different example using light emitting diodes, LED lights. LED light exposure inactivates enzymes, disrupting cell membranes. And that will destroy microorganisms as a result. We can use LED lights for all of these species. Here is the peak length identified for most microorganisms, 425 nanometers. And in addition, what else does LED light provide? It kills microorganisms. It also delays fruit senescence, allowing product to last longer with the least nutritional losses. It also allows for bioactive compounds to be produced, such as phenolic content, anthocyanins, organic acids, vitamin C, for example. When a product is minimally processed, it would be an important factor for marketing efforts. A minimally processed mango has a higher level of vitamin C than a mango that was processed differently. So, since consumers are very focused on quality nutrition, that is an advantage that can be used to sell more product. Here we see research carried out by Kim et al., in which LED effects have been demonstrated certain types of microorganisms. Here we see E. coli. When applied at 405 nanometers, the population was far lower than the control group, which did not have any exposure to LED. Letter B is Listeria monocytogenes. I don't have as dramatic of a reduction as with example A, but it is a significant drop nonetheless. And example C with Salmonella, we noticed that reduction reached 0, which makes it undeniable that LED exposure would be beneficial to human health. Care was taken in terms of cleaning equipment, operator handling, and still microorganisms were present. So, what is the advantage of LED lights? microbial resistance is not induced, and that is something quite relevant these days. The more chemicals applied to the product, the more induced resistance will happen. It is non-hazardous because there's no mercury involved, low radiant heat emissions, compact size. And what is the consequence? If you allow for a microorganism due to heat in the water penetrates the fruit, you will not be able to reach this sort of efficiency in eliminating them. Another option is the use of cold plasma. Cold plasma is the fourth state of matter. I don't know if many of you have heard of cold plasma. We're very familiar with the solid, gaseous, and liquid states. However, when I submit a product to great pressure or change in temperature or electromagnetic field, I generate a cloud of free radicals, protons, anions, as we see in this diagram. It is a mix of compounds which are scattered. They are apart from the main content. I only managed to find an illustration showing a meat product. However, whatever the product may be, when it goes through a cold plasma current, those ions, protons, anions react and degrade microorganisms. And here we can see in greater detail what exactly happens as those microbial cells go through that cloud of products. What happens is nucleic acid cleavage. The microorganism is degraded, cell membrane rupture, protein is denatured, lipids are oxidated. And so, as an advantage, we see that there are little to no changes on nutritional value or any other physical characteristics. It's low energy and it's quite safe. Going into greater detail, we will see the total level of microorganisms when I apply cold plasma in comparison to a control group. And after only 4 days, we see greater level of microorganism control. When I consider firmness of product, look how interesting. Cold plasma not only destroys microorganisms, but it allowed for product to retain its firmness, which means potentially longer shelf lives.
A level of sugar also higher that makes the product more firm and sweeter. But in this research, they weren't able to create an adjustment to not increase acidity. So even increasing a sugar, the relationship between sugar and acidity that reflects flavor shows that the flavor worsened slightly than compared to the control. So repeating what I mentioned since the beginning of the presentation, the need to invest in research to make sure that the product maintains sensorial quality. The next is water. The neutral electrolyzed water and acid electrolyzed water both destroy bacteria and fungi. And bacteria destroys DNA and the fungi oxidizes the cell wall. How does this work? The majority of people already heard about water pH change to make water healthier. How does this happen? It's going through an electric current in a water solution. If I put an anode or a cathode, I can produce particles in water. And if it is a cationic water, I produce hydrogen. And if it's ionic, I produce oxygen. To a reaction, two molecules of water plus produce hydrogen plus hydroxyl. And if I place two currents, two molecules O H2O, that it produces water plus the electrodes. In if we this will create a pH 6 to 9. That's why it's called neutral. However, we cannot it doesn't stay in neutrality for a long time. It reacts again and becomes again a water without neutralization. This is reduced in the presence of organic compounds. The electrolyzed acidic water, it will act in a pH of 2 to 4. So this is the treatment pH. And then it does not lose is the quick power. It can be stored. And due to its acidity, it causes corrosion of equipment. The improvements equipment. So there's few practical applications for this method. It has a potential because it does not cause residue, but there's still issues that need to be studied in order to see how this water is not corrosive or it doesn't lose its potential to lose its properties quickly. Here we can see the effects of electrolyzed water, acidic or neutral, in mango. In diverse products, what do we understand? We see that for the darker bar here, it was able to create a reduction of microorganisms much higher than what I would be able to do with distilled water or with hypochlorite, because this is the potential of microorganisms without treatment, distilled or hypochlorite, and see how both acidic and neutralized water increased the reduction of microorganisms. Certainly, some other methods were lower than the acidic or neutral water. UV light, for instance, showed to be even better, showing that we still need to study further the electrolyzed water for application in mango. This is a drawing showing how to perform this treatment with electrolytes. So I can produce alkaline or acidic water. I use the minimally processed mango and I test how concentrations of electrolyzed water, neutral electrolyzed water on my product, and what results did we obtain with a storage of 6 days and with storage of 12 days. What can we understand is that the neutral electrolyzed water enabled the loss of weight, in this case the blue bar, is lower than the loss of weight in this side. In the left side was similar, and on the right one was less than what we would lose in the control group, showing that in addition to control microorganisms, it reduces the loss of weight in my product. Noticing that in addition to reducing weight loss, what did it do to microorganisms? We see that regarding the control that I have here, in the control group, I had 3.1 10 cube of microorganisms, and after applying the neutral electrolyzed water, it was reduced to 1.5, which is half, simply with the use of water without losing firmness and control, showing the great potential if we invest further to improve this technique. And the last technique that we will see is the high intensity ultrasound, or HIU. Ultrasound is applied with frequencies of 20 to 100 kilohertz and can be, or it can be applied in low or high frequency from 10 to 1000 watts per square centimeter. What does this cause to my product? Similar to the light, it disrupts the cell wall and it causes fragmentation of my microorganism, and this results in the inhibition in terms of injuries to the product. So this is a research with Kent Mango. They saw that the index of decay when treated with ultrasound was lower than for a period of 7 days was lower, and when was treated with ultrasound, much lower than any other applied treatment. Stored in 4 Celsius for 14 days. Going beyond the minimum, which was 10 to store, confirming the potential of ultrasound for treatment of mango. And when we noticed the existence of unpleasant odors, it was also less when stored for 14 days than the control with any other treatment. In this table, we see that the high intensity ultrasound treatment, the numbers are very small, but I needed to show you the full table. So you could compare the starting level at 0 bacteria or 0 storage days was 2.94. When I apply the ultrasound, I have a reduction of the variables at 7 days, 3.40 non-treated, 220 for the treated. 14 days, 3.85 non-treated, 265 for treated, showing the control of bacteria as well as fungi control, which is the table on the right. When I see that for 7 days with control 322, for 14 days, 7 days for treated decreased to 220, and 14 days from 389 went to 272, proving that the ultrasound controls microorganisms in addition to reducing the development of odors and maintaining firmness. Taking into account the amount of information, this presentation was only a thought-provoking presentation so that we can invest in research so the industry can have more available information to produce quality mango and satisfy the consumer demand, not only in terms of sensorial quality of the product, but as well as food safety. So we can see that we know very little about very potential methods that can be used by the industry. I would like to thank everyone for participating, and I'm available for any questions. Thank you, Dr. Francine Kukel. We have a question from Emiliano Martinez. The application of soluble calcium in a washed mango, is it adequate? Yes, it is. It will enable more firmness during the processing, as we showed here. A second question. Good afternoon. What cultivating practices reduce the contamination potential of the products that will be sent to the industry? To have a bathroom on the field, to train the farmers, to tell them to wash their hands, to have paper towels on the field because the cloth towels on the field is a vehicle for transmission of bacteria and microorganism, mainly the Sclerecia, to wash the peel carefully at every harvest, not to put any box on the floor. There are many measures where the producers have to focus and the workers need to be trained. This should be a law for the producers so that can produce a low contamination product that doesn't get to the industry with high levels of contamination. Because if it is highly contaminated, not even these practices will reduce to the recommended levels for the final consumer. One more question, and how does the light applied post-harvest can improve the quality of a product, of a minimally processed product? I have a slide here. I hope that a question like that was asked. Just to briefly explain that to you, we all know that the light on the field will produce protein, it will produce nitrates, phenolic compounds. It will control diseases. In this way, we understand that light has an effect that induces the production of compounds. How? By acting on the genes. So it stimulates the activation or deactivation in the promoters region. So it would be reasonable to understand or presuppose that if the light has morphogenic effects in the field, it can also have photomorphogenic effects in the harvested product. The harvested product has chemical reactions that are synthesized with products that were not on the field at the expense of its own reserves. So this does not mean that a product harvested with a certain level of vitamin C on the field cannot increase the vitamin C. How? Applying light on the belt that converts these ROS species that will actually activate the production of many compounds. It increases the quality with the action of the genic expression of what is already on the product. In a complex way, it acts on the product with this gene expression and produces innumerous chemical reactions, changing the conditions and the quality. With these changes, we increase nutritional quality and quantity, increased firmness of the product, and many products that are substances that are interesting for the industry itself. Perfect. One more question from Luis Figueroa. The utilization of microorganisms or beneficial matter with frequent use as one time month can help or not the matter and or microorganisms that we call smart. What do? They, when applied, make the mango produce substances that will enable the improvement products that improve the quality of the product. As a general example, if I want to have a higher a higher production of the factors that increase the color of the product, this will increase the better coloring of the product because the consumer seeks mangoes with better color. So it's an advantage to improve the quality of the product. It's highly recommended.
Let's have one last question. We have a question from Pedro Mariotto, and the question is, adding calcium through the soil can be used as a viable strategy to help obtain higher quality fruit for processing? Absolutely, it is essential to study calcium for each orchard, for each cultivar, the time of year, the amount, and the vehicle through which it is applied so that the cell wall can be stronger and firmer, and the calcium will allow the fruit to become more resistant to all processing. A calcium deficient fruit will not withstand processing as well. Very well. Thank you once again, Dr. Francine Coquille. We hope to see you in future meetings. The next meeting will be July 11th at 3 p.m. Eastern Time, and we will talk about symptoms and management for mango production. Thank you so much.